Hello, this is just a tester for the video. Everything seems to be working, and I think the speed is looking good. So at this point, you'd probably just be an introduction of some sort, something about wing patterning. I don't really know. I got a picture of a Drosophila in case you want to talk about Drosophila. Yep, everything's good. That's just a picture of the wing. I don't know why, but you can make up something. Okay, development of the Drosophila. So at this point we have a bit of a timeline going on. So the egg is fertilized, and then pupation and growth, and then I'm a bit ahead of the video, but metamorphosis, and then the adult stage. And you'd probably want to make it clear that we're talking about more the pupation and growth stage, because that's kind of where the segmentation and polarity occurs, which is kind of the point of our entire thing. Imaginal disc. Right, so how do we get from this to this? So from the sort of the imaginal disc stage to the wing stage. I don't know what you're going to talk about, but just write something. Morphogens, that's quite a crudely drawn imaginal disc. So you might want to mention that the imaginal disc is actually just like a sac of cells, so it's kind of like a round thing, and then eventually when the wing forms, it's flattened. So here I've just drawn the different morphogens, so starting with engrailed, which is expressed in the posterior compartment, and then hedgehog, which is expressed from posterior to anterior, decapentaplegic, which is expressed along a strip in the middle, and then the gradient occurs like going outwards from the middle, and then finally wingless, which is secreted sort of the other way, or along the sort of proximal distal kind of margin. And then as well I've sort of included notch and apterus, which are some other ones that were on the wiki page, but I don't really know what they do. I guess you don't really need to talk about them very much. And here I've sort of said that, well, all four of these genes sort of set up the wing so that they're split into their compartments: anterior, posterior, ventral, dorsal, proximal, distal, etc. So that the imaginal disc just flattens out and becomes that. Now here, of course, all of these genes, not only do they work together to create segmentation, but they also can follow along and have their own signaling cascade, which will result in transcription of different genes that will lead to the patterns that you can see in the video there. So those are some of the different patterns that you can get. Drosophila melanogaster in particular doesn't have very many patterns, but like other species of Drosophila that use sort of similar kind of mechanisms do. So that's just the three things that are affected, and then a bit of a statement about pattern formation, and then I've sort of done a quick kind of, so that's kind of the whole thing there stretched out, and then credits at the end. So yep, have fun.